Hi, everyone. I hope you're all as excited as I am for another intriguing addition to our monthly Microbial Insights webinar series. I'm your host, Dexter Puckett, project leader at Microbial Insights. And once again, we are grateful to have with us another skilled and experienced expert in the field, Mr. Isaac Pelz. Isaac's presentation, titled Effectiveness of the In Situ Bioremediation of a 1,2 DCA plume in Saline Groundwater, will be a methodical start-to-finish journey into a fascinating case study example. Over the years, Microbial Insights has maintained a wonderful relationship with environmental resources management. Top to bottom, our experiences working with them over the years have been overwhelmingly positive. It is so great to see our molecular biological tools being applied to generate actionable data. We are thrilled to have ERM presenting for us today and look forward to what the future has in store for our companies. Isaac Pelz is a principal consultant at Environmental Resources Management. He specializes in the characterization and cleanup of contaminated sites to support closure and redevelopment. He holds a Bachelor's of Science in Geology from California State University, Sacramento. He is a licensed professional geologist and a certified hydrogeologist in the state of California. As many of you have attended before know, this webinar today will be recorded and available on the resource section of the Microbial Insights website within the next week. A link will also be emailed to all registrants as soon as it is available. At the end of this morning's webinar, there will be a question and answer period. Please type your questions in the GoToWebinar software and our speaker will answer them at the end of the presentation. You can also email any additional questions to info@microbe.com. All right, Isaac, it's all yours.
All right, thanks, Dexter. I really appreciate the opportunity to present today. So thank you to Microbial Insights for hosting, and also thank you to everybody who's joining. I know we're all very busy, so attending it means a lot. So with that, I hope the presentation is clear and everybody learned something. So this morning, I'm going to present on a field pilot test where we completed an evaluation of whether bioremediation would be effective for 1,2 DCA in saline groundwater. And the backstory here is that originally when we wanted to evaluate remediation in saline groundwater, some of our subject matter experts expressed a lot of caution that the higher salinity could be problematic for remediation. And so I just thought, all that we've learned from this, it'd be worth sharing with all of you about how we approach this, how we designed our pilot study, and some of the things we learned. And also just want to say, a lot of the tools that we learned for this pilot test, came from industry presentations and Battelle and other things like that. So it's a bit of giving back, just recognizing we all kind of contribute to each other and best practices in our industry. And just one more word before I dig in. I think this is a timely topic because a lot of the remaining cleanup sites are more complex and challenging, and also because with the topic of sea level rise and seawater intrusion, that's what I want to say. This topic comes up a lot where it's a question, hey, can we treat this, this groundwater that's impacted with salt? And so I thought this would be timely and relevant. So hopefully everybody can learn something, like I said. So in this presentation, I'm going to aim to answer the following questions. How can elevated salinity affect or impact in situ biological reduction of ESCs? So what are the processes? What is inhibition about? How can it affect sites? So a little bit about, not a microbiologist, but I'll give it a swing. And then second, how is our pilot study designed to overcome and evaluate its effect, specifically to answer that question, and in regards to a 1,2 DCA plume. I'm also going to go over the pilot study results to dig in and see what we learned. And then, with those results, I want to provide as much insight to what we could apply at other sites. So all of you, if you have your own sites, what could you learn? And real briefly, I've used the term bioremediation and in situ biological reduction. I'm not going to go into great detail about some of the basics of these processes, but I want to give just enough so that those who don't delve deeply can kind of understand what I'm following. So in situ biological reduction is really using microbes to break down contaminants in groundwater. And the in situ part is doing it in the subsurface. So you're injecting in and it's happening underground. You're not pulling anything out and then treating it or something like that. And the process, biological reduction, again, it's reducing or anaerobic conditions where the microbes, their cellular function will break down contaminants and destroy them. And it generally includes removing the chloride or the chlorine atoms from contaminants, in this case 1,2-dichloroethane, and replacing them with hydrogen, and it results in them being converted to non-toxic end products or inert end products. So it has a lot of advantages over different technologies. So here's a quick outline of what I'm going to present today. Firstly, like I said, I'm going to talk through some of the site conditions and also about what salinity and inhibition and what is that. Then I'm going to get into the pilot study design in the second section, setup, implementation, and monitoring, a bit about how to set up pilot studies so that they're effective and they can answer the questions and the objectives that you set out. Third, I'm going to go through the pilot study results and dig in and see what we can learn about that at this site. And then lastly, I'll go through conclusions and lessons learned, and then some more recent next steps for our particular project. So the background here is that this site is a former chemical manufacturing plant. And for this particular part of the site, this impacted area, they manufactured ethylene dibromide. So it's a pesticide. And really the contaminant of concern, like I said, is 1,2-dichloroethane. And so we're not exactly sure how the 1,2-DCA is there. Whereas EDB was manufactured, it might have been a co-product of the manufacturing process, but that's really speculation. And so as you can see, the site that's a drone photo of the site area. As you can see, it's in a coastal environment. Hence some of the seawater intrusion that we'll talk about. And the site's undergoing redevelopment. So you can see some of those structures being built in the vicinity of the site to the left there. And so some of that relates to the site cleanup and moving on and kind of recycling the land into a more productive use, which is something that's very sustainable, and we're really proud of our work at ERM. In addition, the site has had a pump and treat system operating in this particular impacted zone. It's been operating since the mid-'80s. So we have a lot of sites with these legacy pump and treat systems that maybe they were effective once upon a time. Maybe it was one of the only technologies that was effective once upon a time. But as time goes on, their effective diminishes, and the time frame to achieve cleanup goals can be very long, as most of us know. So in addition to the pump and treat system there, there's been impacts down into the deeper groundwater, like I said, and the deeper groundwater has also been impacted by seawater intrusion. So this happened, maybe the 70s and earlier, where there was a lot of agricultural activities in this area, and there was overdraft in the basin, and then there was seawater intrusion. So you kind of have that kind of inland migration of seawater, and you also have kind of a downward release of contaminants there. And so at this site, obviously we're evaluating if in situ biological reduction can be an alternative to pump and treat. So on the right side, you can see kind of a quick cross section of the area we're talking about. So again, this is 55 to 65 feet, although it varies, maybe as much as 40 to 70 feet. So this zone we're talking about is this kind of pale yellow down there at depth, and there's just a few, concentrations from our characterization on there. Yeah, so again, we set out to ask the question, can we implement a more sustainable remedial action to replace pump and treat? And further, can we use in situ biological reduction to do that? And so I'll continue with how do we later on I'll talk about how do we get to in situ biological reduction as opposed to other technologies, and also some of the other aspects of biological inhibition of salinity and what that is. Here's a little bit of background information about the groundwater and what's in the groundwater in our treatment area. So as discussed, this is a coastal cleanup site that's primarily impacted with 1,2-DCA, and you could see there, about 1,000 micrograms per liter currently remaining. So kind of modest concentrations in the scheme of things. There's also some trace concentrations of other contaminants, as shown, TCE, EDB, and chloroform. So in terms of the actual chemistry and the salinity, what's in there, you can see some of those numbers on the screen on the left side there. TDS and chloride are the typical, two kind of easy to wrap your arms around for how much salinity is there. You could also look at conductivity as a field parameter, and just you can convert that back and forth to get a handle on what you're looking at there. So I wanted to kind of put up the salinity numbers in context. So the graph on the right shows how our site water compares to seawater, and then kind of fresh water at the site that doesn't have seawater intrusion. And so start with the red bars there. That is chloride and TDS respectively, and what they are in seawater. So that's generally seawater. It varies, but that's kind of what you're looking at. And then on site, we have about the blue bars there, 7,500 and about 15,000 milligrams per liter of chloride and TDS. And then the green is kind of site fresh groundwater that we tested, and it's pretty fresh, right? It doesn't have very much in it. So ultimately, we're looking at some kind of a even mixture of seawater and fresh water, and other things in the water kind of help fingerprint it. Bromide is like 80 milligrams per liter, something like that. So that's often a good indicator of where did the water come from, what's kind of the makeup of the water. Very rough. In addition, there's a fair amount of sulfate, which again is often typical of seawater. Most fresh groundwater doesn't have that much sulfate unless there's some kind of interesting geological condition. So 600 milligrams per liter, and that's I'm going to get to that later a little bit more, but that's not typically something that's going to inhibit biological remediation or biological reduction. It's just something that's going to demand more organic carbon. So when you're dosing your reagent, something that's really, really important, probably the most important parameter that we look when we're designing, biological reduction remediation. There's also neutral pH and other things that you might want to look at if you're looking at feasibility of these studies. So here, like I said, I'm not a microbiologist, but I can dabble. So we'll get into a little bit of what is salinity inhibition. And so we know that elevated salinity can kill, or it could slow microbial activity for certain types of microbes. And certain microbes can tolerate different levels of salinity. Basically, they are adapted or they are conditioned or to tolerate certain levels of salinity, and there's something about the salinity outside the cell wall and inside the cell wall. And if they can't tolerate that range of salinity, then there's going to be pressures and gradients of the water between the outside and inside the cell walls. And so you can see over on the right side, really, of this cartoon, that if the salinity on the outside is very high, but the microbes need lower salinity on the inside, then that gradient can cause the microbes to shrivel up and potentially die, because they're always trying to achieve an equilibrium between those cell walls. And so again, that can result in lysis, cell destruction, or just really slow growth. We were told that it's something like trying to have living things in a desert, right? Maybe they'll survive, but they're just going to scratch out a living. So anyway, the right side is like a visual of what it is when there is high salinity and the microbes just are not able to function in that. And then the right side here is this microscopic view of what's going on. So hopefully I did that justice to the microbiologists on the call. And so when we reached out to some labs and said, well, what kind of number should we look for if we're interested in how much salinity is too much for biological reduction, bioremediation? We got some answers that were focused. One, they were focused on TCE remediation, because that's really the most common one we're looking at, TCE plumes. Two, they were focused on dihalococoides, which most of us all know. It's kind of the microbe, the one that is known to dechlorinate TCE all the way to ethene. So the one. However, our site isn't TCE, it's 1,2-DCA primarily. And what about other microbes, right? DHC, it might be a little more picky. So there's some unanswered questions there. We did get some kind of rules of thumb about what levels of salinity are an issue and which ones aren't. So ultimately, at sites with higher salinity, you want to do a bench study or a pilot test to determine if it's an issue at your site. So it's kind of just another step to add. It's often a wise step rather than going full scale or something, and then you find out you have a problem. And so, like I said, this is also what our kind of subject matter experts said. They're like, we're really skeptical. They've had some horror stories, quite a long time ago about, implementing some remediation, and then there's salinity inhibition issues. So, with that, we'll really get into selection and design of the pilot study. So firstly, why are we talking about in situ biological reduction and not other remediation activities off the bat? And the reason is we've used in ISBR at this site in shallow water very successfully. So, this wasn't just something we pulled out of the bag and so let's try this. We already knew that it works on 1,2-DCA and the non-saline groundwater at the site, and very successfully. You can see on the right side there, that graph, those are the 1,2-DCA remediation results for shallow groundwater injections. And, similar concentrations, same COC. At that time, we weren't sure, is 1,2-DCA, is it going to be conducive? We're going to have problems, but it was, that's about as good as it gets, right? So that really declined quickly. We didn't see rebound. And so that's where we started. You want to build off of what rather than trying to reinvent the wheel, right? And just to address a couple of other technologies that might work, you might be wondering, does ZVI work? Does chemical reduction work for 1,2-DCA? We understand it does not, based on our research. So we weren't looking at ZVI, nor we think does biogeochemical processes like sulfadated irons for 1,2-DCA anyway. So it probably is possible to do ISCO, but it might be some further evaluation needed, but you might need a lot of reagent to get out of over a high soil oxidant demand. So with that, I'll keep going and talk about site background. So looking at the treatment zone conditions to further evaluate if injection-based remediation is going to work, we wanted to dig in really to the hydraulics of, can we get the reagent distributed in the subsurface? So like I said, the treatment zone is about 55 to 65 feet. It varies a little bit, but it's quite sandy. You can see a picture there of a soil core. A lot of it looks like beach sand, and it's fairly homogeneous and laterally continuous. Really good conditions for injections. In addition, the treatment zone is bounded above and below by fairly competent aquatards. As you saw, I think on that cartoon above, you can see the aquatards above and below. You can see it here too in this kind of snapshot of one of our boring logs. You have lean clay, above, and then it's below. These are two kind of well-known aquatards that are present pretty much everywhere in this area. That are nice bounding units for when you inject a fluid in between here, it kind of stays put. So that all checks out. So here I'll get into the pilot study approach. So like I said, we recognized we needed either a bench test or a pilot test before investing a lot of capital into remediating this deeper aquifer here. We did look into a bench test, but biological reduction, it's not a particularly fast process. It could take a few months, and so there's a time commitment with the bench test, and also there's a cost commitment. With labs running it, taking some time. There's certainly advantages to a bench test, but ultimately we went with a pilot test because it was a fair amount of expense for a bench test, and with not too much more money, we could do a pilot test. We can actually test it in the field, in situ, and get maybe a little more realistic data. We could also hopefully maybe get some remediation done while we're doing it and kind of add some value. So we went with a pilot test in the field. And for the overall approach, we installed 2 wells 10 feet apart. And so really thankful that we got funding to do this rather than try to use old wells that are maybe not screened right, or who knows what their history is, and try to recycle them. We got to install two wells, properly designed and constructed with sonic drilling, so that we could get complete soil cores to make sure we set the wells right. We also did groundwater sampling with sonic drilling while we're installing them, so we could, again, further make sure that we're injecting into the impacted zone. And again, 10 feet apart, I think it's a critical design factor, because you want to get your reagent injected close enough that you have, very good distribution in your monitoring well to get good results. If you don't get it into the monitoring well, you can't monitor it. It's failure of the pilot test design. So as you can see there, we have an injection well and a monitoring well 10 feet apart. We'll talk about getting the reagent injected into the injection well later, but definitely want to have enough that you get a complete ROI and radius of influence to get into your monitoring well. We also made sure we selected the pilot test to occur in an area that was representative of the overall plume. You don't want to just pick a place where the salinity is kind of low and then it's not representative and you can't apply your results to the kind of full-scale treatment area. So we made sure of that. And again, like I said before, I think we wanted to isolate other variables to just test effectiveness. So that's why you've got to get your reagent into that monitoring well and get good distribution. That would be so you're only testing for effectiveness, right? You're trying to focus the question in for a pilot test and just answer kind of one question. If you can get other data, that's great, but really you've got to answer that one question to start off. You want to talk about the reagent that we used, and to start off, we really needed to use reagents that we could easily inject into wells, into well screens. And I forgot to mention that, yeah, we made sure we installed the proper well screens for injections. So we've got a nice, open slot, 40 slot wire wrap screen, which is really nice for injecting and really as big as you can get it to for injection efficiency and just get the reagent without any trouble. But yeah, that's really a design factor. You got to use reagent that you can easily inject and distribute into a well, so that some of them might be crossed off the list, but those are more chemical reduction reagents like CVI that might be a little more tricky. But anyway, some of these they're all easy to kind of mix and inject. So first thing was we wanted to condition the potable water we got to make sure there's no dissolved oxygen or other things that could be harmful to the microbes that we were going to add to the water for bioaugmentation. So we used Newman's Own OS oxygen scavenger to get that water to be anoxic to start for a mixed water. For our carbon substrate, we selected Evonik's ELS product there. So that comes in 100% strength. It's some thick stuff, so it's a little bit cheaper to ship. And then you just have to make sure that you can shear it out when you're mixing it. But yeah, we've used ELS in several projects. We like it. We also used EHC liquid, which is really, it's similar to ELS. It has carbon substrate, but it also has iron salt. So the excess iron can be good for a few things. It can help you, further achieve reducing conditions with the ferrous iron, converts to ferric, and can also be a supply for when sulfate is reduced to sulfide. You can get more iron sulfates and lower the amount of hydrogen sulfate that you may generate. There's a few other benefits, but we also added that, and that's a point here. You want to kind of throw in the kitchen sink here in the pilot test. You want to, get your best shot, put a bunch of things in and have your best chance of success. So that was part of the EHC liquid. Really critical for this is we bioaugmented. So that means we, directly added microbes to the reagent here with kind of a diverse suite of microbes. And this was something that we learned from the shallow treatment that we did. Not just adding Dihalococoides, DHC there, but also a bunch of other microbes in MDB1 like Dihalobacter and Dihalogenemonas and things like that. That was a lesson that we'll kind of go through a little bit later here about that being critical. We think that some of those in MDB1 are really doing the heavy lifting here. So we did add the Dihalococoides, and that's I think Cyrum's KB1 Plus product, which is supposed to be more tolerant of pH ranges. So we're thinking, well, maybe that'll help with salinity too. So let's try that. So we also added a pH buffer, potassium bicarbonate, because you can often get pH drops with microbial activity, which can then be toxic to the microbes. And we added a fluorescein tracer dye, so that we could easily see the reagent. We'd have like another way to track its appearance when we inject it. Some of the injection details are here. We injected almost 5,000 gallons of fluid into our injection well. And when we look at what is that in terms of mobile porosity, that's about 18% when you're looking at a 12-foot ROI. So that's a lot. Like in my experience, that's a lot. And the backstory is, we probably got more like a 15-foot ROI or something, which equates to a lower mobile porosity. But we didn't have a way to track it beyond the monitoring well, which is 10 feet away. We could have put another well in, but they're a little pricey. So, but this is definitely adequate for a pilot test. Yeah, then 920 pounds of ELS, that's 2 drums, which is a little over 2%. Again, that's probably more than we needed. But for a pilot test, you really want to make sure you don't underdose it. So, and then, yeah, we injected at a lower, modest flow rate, 4 GPM at pretty low back pressure. We didn't really want to, crank it up and test higher pressures. We wanted to just make sure we didn't risk, breaking any seals in the well or in the aquifer. Really just get it in easy. Although this flow and pressure, it did show that we could probably really crank it up with no trouble. Which, by the way, we did during a subsequent pilot test, and it was a very good well. So here's some photos of the setup. Here's the manifold injecting into the well. Here's the mixed reagent. So it's a nice microemulsion of oils, and then some of the mixed tanks there. So the real key part of the pilot test is monitoring to make sure that during and after that you inject, that you're getting the data you need to tell a compelling story, to answer your questions and make sure that stakeholders can see that they agree with you, that what were the conclusions. And so the monitoring during injections, we were able to actually pump at a low flow rate from our monitoring well in real time to look at where is the reagent showing up during injections, which is really great. We were able to actually put it through a flow cell and get geochemical results. And then we could also look at the tracer dye. You could see there's fluorescence here. We got tracer coming through in the purge water. We also got TOC results. So that was really helpful to know the appearance of the reagent when we inject it here to the left. We saw it show up over here. And an interesting side note I wanted to share was that we could actually see the drop in conductivity, we think, as the reagent showed up in the monitoring well. I thought this is really cool to look at, but the conductivity in the native groundwater is quite high. It's saline. And then as we see the reagent show up, which is very quickly after start injecting, we saw the conductivity drop. And then overnight, when we were injecting over the weekend, it kind of returned. And then so anyway, this is just a cool signal to kind of see. So there's various conclusions you can draw from that. And then after injections, we did monthly groundwater sampling, which is maybe more frequent than you need, but it was helpful to really quantify when we're starting to see the effects of the remediation, which we'll get into a little bit later. We'll get into it now, in fact. So, the bottom line is here, it worked really well. There's a lot of information on this slide, but the treatment worked very successfully. And so, we saw significant reductions in 1,2-DCA, in a few months, basically. We went from about 1,000 down to the low ones, which was really great news. So it's the green line on the graph over here is the 1,2-DCA. So we got some testing immediately after injections, but really the 1 month data is over here at this. You could see my cursor. So yeah, we saw reductions in our 1,2-DCA. We also saw some reductions in the other constituents, which was good news. And critical also, you're always looking at rebound and you're curious about rebound. We didn't see rebound at all over 11 months of monitoring. And that's really great news. Sometimes it's hard to prevent rebound, but in this case, it may suggest that there's not a lot of sorbed mass in the aquifer that's gonna kind of back diffuse in. Also, yeah, 1,2-DCA is very soluble, so maybe there's less of that anyway. But it was great that we didn't see rebound nonetheless. And then in terms of the geochemistry, these graphs on the right are set on the same x-axis. So you can really see, digging in more to the results, we had a nice spike in TOC, this red here, quite a lot. That's where I typically want to see it, around 1,000 milligrams per liter, and then it kind of decayed as this really bug food is being consumed, right? And then the resulting effect is sulfate reduction, which I really would like to see sulfate reduction in this case, and we saw it very quickly. 1 month out, and we're at sulfate reducing conditions, basically. So these are all good geochemical indicators that would then suggest the onset of reductive dechlorination. Another great thing about 1,2-DCA, I guess I could say, is that we saw generation of chloroethane, which is a breakdown product of 1,2-DCA. And that's just a great indicator to know that you're not just diluting out your plume, you're actually destroying it. So the chloroethane is, I think you pluck one chlorine off and you add a hydrogen or something like that, and you can convert 1,2-DCA to chloroethane. So we saw that appear, which is again good good indicator that you actually have a breakdown. We also didn't see vinyl chloride, which is great news. As I'll show in a second, that could be a pathway for 1,2-DCA destruction. So here's a slide on what happened to the 1,2-DCA. So the graph on the right shows the DCA and its degradation products in molar units. So they're kind of 1-to-1 in terms of moles. So you can kind of directly compare what's happening to the 1,2-DCA. So 1,2-DCA drops, this green line, and then you have the appearance of chloroethane. So this, in terms of bulk magnitude, about a third of the 1,2-DCA was converted to chloroethane, right? Based on the height of these graphs. And then you also see some ethane generated. So that's, looking at this, I kind of took some flow charts I found. I squirreled away some of these breakdown flow charts and to kind of see what's going on here. So you have a lot of this chloroethane reductive dechlorination, and evidently you have some ethene dehaloelimination, and ultimately we did see a ton of CO2 and methane, but you can get all kinds of things happening down here that we may not be able to detect. We may get conversion rapidly to CO2, then you never even see it. So anyway, this may underrepresent some of the pathways that we're seeing here. That was kind of cool to see. Something that was really helpful that we were able to do was actually quantify the microbial populations in the groundwater before and after our pilot test. So this slide shows some of the results from the Microbial Insights Quantarray Chlor to that end. And we are aware that there's many microbes that can dechlorinate 1,2-DCA. Some of these are kind of fun to say, the halogeninomus. And so we knew that, and we were looking for them. We actually injected these microbes, right? DHC, DHG, DSB. There's, I think, the Halobium chlorocercia, not showing off. I spent too much time trying to pronounce these. So we injected these, monitored before, that's the green bar, and then we monitored after, that's the blue and the red. And you could see there's a massive increases in the microbes after injections. This is, I think, 1 month and 4 months after. So massive increase. And it's really in these microbes that are other than DHC. So we're talking orders of magnitude. This is a log scale. So massive increase in these other microbes besides DHC. And then a note, we never saw DHC rise up to a level that was useful. According to some research, 10 to the 4 or 1 to the 4. It was always a little bit low. So, not sure if that was some competition from the other microbes or it just wasn't happy. But anyway, the other microbes, very happy. So we could conclude that these other microbes, some of them were the ones that were doing the heavy lifting here, dechlorinating 1,2-DCA. So some of the other geochemistry that's interesting, I'm not going to claim to be the best interpreter of these results, but maybe others can. Just some facts that we saw. CO2 massively increased. Yeah, CO2 is this orange bar, right after injection. So that's an indicator of a lot of biological activity, although it may not all be useful to dechlorination. And we also saw iron increase. Ferrous iron, as you would expect, increases. If not because you're adding ferrous iron, because you're getting reducing conditions, and then you just kind of free it up in the aquifer matrix. And then, like I said, we saw sulfate, drop down to really completely reduced out in about a month. That's this one here, the blue. And then methane increased, pretty massively. A little bit later, 2 months out, it's really here. So that kind of makes sense, right, for your reductive pathways. You get the sulfate reducing, and then you get to methane reducing conditions. We did see a temporary pH drop here. You could see down to 5.4 about. We see this at many sites where we still pH buffer the solution, and it's sometimes we're only doing DHC and it's only TCE we're trying to tackle, and it works fine. So we often see this temporary drop comes back up and no problem. So just a little side note, we see it, but then it kind of recovers, and ultimately it's not catastrophic for the microbial populations, at least those besides DHC in this case. You know, here, like I said, when we do a field pilot test and it's successful, we got something to show for it. And I was really pleased to see that we've kind of nailed a part of the plume here with our pilot test. You can see our pilot test areas over here in this part of the plume, and you know, it's really kind of shrunk it down. We kind of took out a part of it. So just it's nice to see some progress in this plume that's been here for so many decades to be kind of obliterated in a few months. So just to wrap it up, some conclusions. You know, being able to design a pilot test, you know, from scratch with properly designed and spaced wells, and you know, put in the proper reagents really was great to test, you know, is salinity an issue here? And the data that we gained was helpful in helping convince us and the regulators that this was a viable technology. We were able to reduce the 1,2 DCA, you know, by several orders of magnitude. We didn't see any rebound for 11 months after, which is really great news. And it occurred even under saline conditions. So just some of the stats there, if you got your site you're looking at, and in this case it was 1,2 DCA, but anyway, it worked well for these concentrations. We also further confirmed that some of the other microbes out there in MDB1, which I think is applied by Aptum, they may have been the ones that were really doing the work here rather than DHC, just based on the quantarray core results. And you know, if you got a site with difficult geochemical conditions, you might want to consider some of these other ones that, I don't know, maybe they're more robust. Maybe others know more about them than I do. But hopefully these were helpful, you know, tips for others designing pilot tests in these conditions. And then a quick word on next steps. I kind of add this in a little later than when I initially prepared this, because we've made some more progress since this presentation was put together. We did do a phase 2 pilot test to evaluate injection methodologies and scalability, you know, more focusing on what other methods can we use to inject. Can we really crank up the injection rate to have like a feasible, you know, injection at scale? You know, overall that was successful. We learned a lot from that, and then we implemented ISBR as an interim action. We injected a fairly large treatment area in a short time, over 2.3 million gallons in 2.5 months, which might be a good part 2 presentation. And yeah, Cascade helped us with that. Shout out to them. And here's a photo of some of their work with some crazy manifolding for that at the site. And we're planning full-scale injections for another part of the treatment area in 2023 with some recirculation. So that'll be exciting. So that's all I had. Just want to say thank you to the other support folks on the project to make this possible. Yeah, there's other folks here that aren't here that worked really hard on this project. So shout out to everybody else on the team, and especially Dan Lay. Dan gave a really great presentation last week on some of the basics of pilot test design. That was great. But Dan's taught me a lot over the years, so I want to thank him. But that is all I had. Happy to take questions.
All right, well, what an awesome presentation, and thank you for that, Isaac. So if you have questions anymore, please go ahead and enter them into the software, and I will share them for Isaac to answer. So get started. We have some already. So one question I had particularly was, so how far was this site from the coastline? Just curious. I know you're in California.
Yeah, it's half a mile or so, I would say. Yeah, pretty close. And there's also some salt water. What do you call those? Salt ponds, where they would you know, let salt into these ponds and evaporate it, and then they would harvest the salt. I don't know if that had any impact on on this treatment area. But yeah, there's definitely some salty water pretty close by.
Did you see changes in salinity, like when you test, that were really inconsistent? I talked to someone else before, and they were talking about shifts because of the tidal impact. So it's just something I was really curious if you see. He's worked out there also and was saying that there was some tidal impact on the shifts in the salinity.
Yeah, we did not see that. And just a side note, yeah, like you could do a full hydraulic study on tidal shifts. We've done it at many sites, but the tidal impact out here, it's barely measurable. So a backstory is that the local water agency has done a lot to return the natural westward flow of groundwater here. And maybe because of that, you have a nice westward flow. There's more gradient from the east, then there's less impact of tides. So no, we didn't see that, and definitely something to look at and be mindful of, and really, really cool hydraulics if you do see that.
Cool. Okay, another question. Did the chloroethane degrade, and did you see any ethene or ethane? Now, you talked about that, and they were also curious about if the chloroethene is harmful, that generating that can be a harmful product.
The chloroethane did degrade, and it's now, I think, in the 10s, current data in the 10s, but it does seem to be, it's not recalcitrant. And in terms of toxicity, I don't think, it doesn't have an MCL. I can't remember if it has another screen level, but it's fairly high. I think it's low toxicity, but that's my guess. We did see ethene, and
yeah, I saw.
yeah.
Cool. And so do you feel that adding the organisms was necessary? So did you need to bioaugment, or do you think that you could have seen a similar explosion in growth with just right for your target if you just did the full-scale treatment? I mean, instead of bioaugment, you just...
Yeah, sure.
Yeah, so that's something there.
Yeah, so the green bars in the graph back there did show that the native populations of microbes were pretty low and kind of spotty. So I wouldn't recommend not bioaugmenting. I think bioaugmenting was really important to get those populations in there. But yeah, happy to hear others if they have other thoughts.
Cool. Do you think the generated gases, CO2 and methane, increase massively after the injection can be harmful?
Yeah, it's possible in a few ways. I guess I could speculate, and I think if you generate enough CO2, you could start to shift your pH down. So that's kind of why you want to add a pH buffer. There's some applications where generating some methane could be problematic. If you're talking about shallow injections, and you can have some explosive issues. I don't think that's an issue here based on the depth. Those are a couple of things I could think off the top of my head.
Cool. So how much approximately, if you don't want to share, that's fine, but how much did the pilot test cost, and what is the estimate to remediate the entire plume?
Yeah, I don't have those numbers handy on my brain to share. So yeah, I don't want to guesstimate and be way off. But I can say that we did look at the feasibility to replace the pump and treat system, and I think our time to recoup the cost for that is a few years. So in other words, the cost to run the pump and treat, if you replace it with a good, robust ISPER treatment and then another polishing treatment, then your time to return that cost for the client would be a few years.
Okay, so are you concerned at all about, do you think you've covered enough time for rebound? Or, do you think there's any potential you may see rebound at the site?
Personally, I don't think there really rebound is a potential after 11 months. I think the other issue you see eventually is you might see impacted water outside the treatment area flush in and cause some maybe recontamination. That's more what I'd be looking for that late, because I mean, yeah, you wouldn't expect rebound. You'd expect it fairly quickly. I would say within 11 months.
Okay. So, so do you think that Was the effectiveness of the bioremediation enhanced because the injection solution had low salinity and diluted the salinity levels occurring pre-injection?
Yeah, no, that's a great point. We thought about that. So we did see a drop in salinity when we injected, although it wasn't significant, and it returned to native salinity like in a month or two. So you could have a temporary effect where the microbes are a little more happy and stable in the more potable, low salinity water, and that could maybe help their populations. But I mean, nonetheless, yeah, I think the data shows that you have effective treatment, and ultimately, yeah, the salinity returned to pre-injection rapidly, and then you still have your treatment 11 months later. So it may have helped, but I don't think it was like masking the effectiveness. If that makes sense.
Did you look at the TDS and chloride concentration several months after injection?
Yes, yeah, we measured them every single month. So we continue to get a snapshot, and that's where I could say that the salinity returned to native very quickly. Yeah.
Cool. Okay, and then finally, let's see. So someone asked about the methane solubility. So they're asking, did the methane increase to greater than 10,000 milligrams a liter or micrograms per liter? And they're saying that 10,000 milligrams per liter concentration of methane is much higher than the solubility in water. So they're just curious.
I would need to go look at my data. Maybe I have a wrong unit.
Okay.
But yeah, can't answer that right now.
Okay. So I think that's about it for questions. So let's see. So again, thank you, Isaac, for an exciting and informative presentation. And thanks to all the awesome people who were able to join us today. Please look for a follow-up email in the next few days with a link to the recording, as well as a link for a certificate of attendance. That's it for this series. And until we meet again, stay incredible, everyone. Thanks.
Thanks, everybody.